Welcome everyone, Mike Fave here. Today I'm going to go over another study. This is actually quite an interesting one. It's going to be a little bit different than some of the caloric restriction studies. It's going to be covering specifically statins, which are a class of drugs that are very prominently used in today's day and age to lower cholesterol under the guise of the cholesterol hypothesis of heart disease. So I'll kind of cover that a little bit here in the article. So let's jump into it. The title of this study is Statins Stimulate Atherosclerosis and Heart Failure, Pharmacological Mechanisms. So the title in and of itself is quite controversial because these drugs are supposed to be preventing atherosclerosis and helping with heart failure and diabetes and some of these other chronic diseases, or the metabolic syndrome. Syndrome X has it's been termed in different studies in different time periods. And the metabolic syndrome is the obesity, diabetes, heart disease, strokes, that kind of that whole pathology, this disorders of the vascular system and of metabolism. So essentially what they're saying of this article, what this base of the article is, that statins are actually possibly making this worse, at least according to these authors. I'm not going to go through all their names because they're a bit difficult for me to pronounce. So I'm going to start off here with the abstract, and it's going to give you the general gist. So what they say is, we present a perspective that statins can be causative in coronary artery calcification and can function as mitochondrial toxins that impair muscle function in the heart and blood vessels through the depletion of coenzyme Q10 and heme A, and thereby ATP generation. So just in, I guess, a translation of that into more plain English is that the statin drugs can impair energy production at the cell by affecting some important components of the mitochondria. They basically induce a depletion of those important components by, and we'll get into this a little bit later, by decreasing cholesterol production, because some of the intermediates in the cholesterol production pathway are actually quite important overall. As they continue, they say statins inhibit the synthesis of vitamin K2, the cofactor for matrix GLA protein activation, which in turn protects arteries from calcification. So vitamin K2 in the PED sphere, or the bioenergetic sphere, however you want to call it, even in the paleo or Weston A. Price sphere, is extremely important in vascular calcification and functioning in conjunction with vitamin D and vitamin A and maintenance of the bones and proper calcium balance and calcium utilization. Chris Masterjohn is one of the main PhD researchers who's prominent in the sphere, who covers the importance of vitamin K and in the matrix GLA proteins. So statins actually inhibit the synthesis of vitamin K. So in doing so, you don't have enough vitamin K for this production of matrix GLA protein, and then you can essentially, that can lead to, or possibly lead to, calcification of the arteries, which is the part of basically the end stage of this atherosclerosis process. So as they continue, they say statins inhibit the biosynthesis of selenium-containing proteins, one of which is glutathione peroxidase, serving to suppress peroxidative stress. An impairment of selenoprotein biosynthesis may be a factor in congestive heart failure, reminiscent of the dilated cardiomyopathies seen with selenium deficiency. This is also called Keshan disease because it's from a particular area in China. I think it's like Keshan province. So what they conclude in the abstract, at least, is thus the epidemic of heart failure and atherosclerosis that plagues the modern world may paradoxically be aggravated by the pervasive use of statin drugs. And I just to go over this last section here, statins are quite important for the biosynthesis of a certain class of proteins that incorporate the mineral selenium. One of those is the famous one in the hormetic sphere and in the alternative health sphere, but glutathione peroxidase, which is one of the enzymes involved in utilizing glutathione to quell or quench reactive oxygen species production at the cell. So the statins inhibit the production of this protein by impairing some specific components that are necessary for that protein structure. The other thing is there's other selenoproteins besides glutathione peroxidase that can be inhibited or lead to a decreased production with statin use, because those proteins also require some of the components that statins block the production of. And so specifically, if you have a selenium deficiency, which would also lead to a deficiency in these selenoproteins, one of the main symptoms you see with it is a cardiomyopathy, or basically a disruption of the cardiac muscle's function. It basically weakens that cardiac muscle, and it creates a dilated state. So the muscle becomes kind of floppy and loose. And you see this in a selenium deficiency. So the decreased amount of selenial proteins that you see with selenium deficiency, and then the subsequent cardiomyopathy that's developed, may serve as kind of a example of what can happen with a lack of selenial proteins due to the statin use blocking their production. So they're basically covering a lot of the mechanisms that they're going to go into here in this paper, and I'm going to go through as well and discuss with you guys. So I'm going to kind of skip this intro here. They talk about the LDL and HDL, all that good stuff, but they come down here and they zero in on the good and the cholesterol hypothesis. So essentially, the good and bad cholesterol hypothesis is what they kind of in quotes call it here. The general idea there is that LDL cholesterol, or LDL, which is a lipoprotein or a protein that carries lipids or fats, cholesterol, triglycerides, all these types of things. They say that here. It carries it to the peripheral tissues. So it takes it from the liver and it brings it to the tissues. So that is your bad cholesterol, in this theory at least. The HDL takes the cholesterol from the tissues to the liver, and then it allows the cholesterol to be excreted in the bile acids and the feces, or your stool. This is generally seen as a good thing in this cholesterol hypothesis, because the basis behind the cholesterol hypothesis is that atherosclerosis is caused by an excess of cholesterol inside the bloodstream. Basically, just kind of, it's like clogs up the pipes, essentially, it's a very basic idea. It doesn't really, when you visualize it and you think, oh, this cholesterol is going to be, it's just floating around, it's these fatty globules, I'm sure it'll clog up the vasculature. I mean, it's a very, I don't want to be condescending about it, but a very simple or basic way to think about it, especially when you start digging into the biochemistry. So the general idea of I'm just going to lower the cholesterol because cholesterol is what builds up on the side, and that's not necessarily how it works. But in this theory, the idea of, okay, if cholesterol is building up on the side, sure, I just want to lower that cholesterol. So I want to lower LDL and I want a higher HDL, because that HDL, it's bringing the cholesterol from the tissues back to the liver, and then it's excreting it. So we want to get rid of cholesterol, at least in that theory. In reality, as we're going to get through this study, cholesterol and the multiple components that are formed during the creation of cholesterol in the liver, which, and we'll get to in a second, by the way, is the main source of cholesterol production in the body, or the body will produce most of its cholesterol by itself. What you wind up seeing is the cholesterol is extremely important for a multitude of functions, whether that's functioning in the cell membrane, whether that's creating steroid hormones through the mitochondria, whether that's functioning in vitamin K proteins, the selenial proteins, whether that's involved in CoQ10 production inside mitochondria to produce energy generation. So the other thing is cholesterol can actually be converted to steroid hormones in the mitochondria, as well as function as components in the mitochondria, or some of the intermediates in this production can function there. So what they continue to say is they frame it here that HDL is considered good, LDL is considered bad, because HDL brings cholesterol from the tissues to the liver, and then it goes to excretion, and then LDL brings cholesterol from the liver to the tissues, so it would actually be increasing the amount of cholesterol in the body. But there's a caveat here. HDL contains an enzyme called lecithin cholesterol acyltransferase to form cholesterol esters, which is transported to LDL cholesterol by ester transport proteins in the plasma. So even though HDL cholesterol, and there's a lot more that goes into this with the different enzymes and different promoter and gene expressions of different components and whatnot. There's a huge array of things that go on that they're just starting to discover now. But what they're saying is that even though HDL technically can remove cholesterol from the tissues, bring it to the liver, there's still some transport of cholesterol from HDL to LDL through some of these enzymes. And they'll talk about some of these drugs as we get down lower. So as I mentioned before, here they talk about cholesterol being produced largely by the body. So what they say is roughly 1.5 grams of cholesterol is required daily in adults for a variety of essential functions, and zero point 0.3 grams, about half of the ingested cholesterol, can be obtained from 2 eggs plus 100 grams of meat, and the rest, the 1.2 grams, the majority of daily required amount, is biosynthesized in adult tissues. So the adult body largely synthesizes its own cholesterol. The liver is a huge player in that perspective. But overall, the body synthesizes the vast majority of its cholesterol, which is why the idea of targeting that cholesterol production, the body's cholesterol production, was a main point with statins. There's other drugs. There's fibrates. There's niacin. There's a whole host of drugs. A lot of them don't show benefit or drastic benefit with cardiovascular disease. The statins initially were shown, and we'll talk about some of that here, but as they continue, they say cholesterol taken up by HDL is transferred to LDL, which is redistributed to and reused by peripheral tissues. So cholesterol is extremely important to such an extent that the tissues are recycling their cholesterol overall. And if anybody looks at any of the steroid hormone cascades, it's a bit tangential, but the majority of the steroid hormones in the body are indeed produced by cholesterol. So if you want testosterone, if you want pregnenolone, progesterone, DHEA, cortisol, estradiol, etc., aldosterone, then you're going to need an adequate amount of cholesterol. So as they continue, they say recently cholesterol ester transport protein inhibitors were developed. So that's what they were talking about here, where there's enzymes that transfer the cholesterol esters from HDL to LDL. They created drugs that block that process. And so they were effective in lowering the LDL-C to HDL-C ratios, but they were essentially ineffective in preventing coronary heart disease or coronary or congestive heart disease. And let's see how they defined it here. CHD, coronary heart disease. So essentially they have drugs that can decrease the amount of LDL in the blood, which supposedly is leading, this excess of LDL is supposedly leading to this formation of plaques in the body. And then also increase HDL, which is the protein that carries that cholesterol out of the body and puts it to excretion. So they drastically increase the HDL and drastically lower the LDL. So it's great. From that theory, it should be great. But what they're saying is the studies are showing is essentially ineffective in preventing coronary heart disease. So that kind of, and as they'll discuss here, is kind of a nail in the coffin to some extent, or more evidence basically saying that this theory that everyone is currently functioning on, or a large proportion of Western medicine is functioning on, may really not be telling the whole story of what's going on. So as they continue to say, moreover, statins or statins plus other cholesterol-lowering drugs were effective in lowering LDL-C, but were essentially ineffective in preventing coronary heart disease, as will be summarized below. All these observations go against the good cholesterol, bad cholesterol hypothesis, and we should not try to explain the correlation between plasma cholesterol levels and coronary heart disease events based on this hypothesis. So what they're saying is that this basic hypothesis of good cholesterol, bad cholesterol isn't really playing out as how they thought. And when you use drugs like statins or like the cholesterol ester transferase drugs, and you lower the cholesterol in the blood and you change the ratios between HDL and LDL, we're not really seeing those drastic drops in heart disease that the theory would predict. So we should probably look for a different theory overall. The problem with this from a industry perspective is that if you've built up a large amount of drugs and research supporting this or that go along with this hypothesis and that lower that cholesterol, well, you can't. It's quite difficult from a monetary or investment perspective to then say, oh, this hypothesis isn't really working, and well, these drugs aren't giving us the effects they want, so let's kind of remove them from the market. But that's an entirely different story, although it does lead into the next piece here, discussing some of the problems around the statin research. So the authors go into, and they say, since the introduction of statins to clinical medicine in 1987, several kinds of statins were reported to be effective in lowering LDL-C and also preventing coronary heart disease events. However, and they say in parentheses here, mostly in the 1990s. However, unfair and unethical problems were associated with clinical trials reported by industry-supported scientists and new penal regulations on clinical trials came into effect in 2004. So after 2004 to 2005, all clinical trials performed by scientists relatively free of conflict of interest with pharmaceutical industries reported that statins were effective in lowering LDL-C, but no significant beneficial effects were observed for the prevention of coronary heart disease. Currently, the majority of scientists continue to claim that statins are effective in preventing coronary heart disease, but these claims are based on meta-analysis reports, including those published before the EU regulation. So those studies were mostly in the 1990s. So what happened was the industry was supporting scientists who were creating a ton of statin studies showing all this benefit, so severe conflict of interest there. And basically the regulatory bodies were like, look, we can't have this going on. So they put in new regulations. Those new regulations came in, it looks like they came in 2004. And then once those regulations came in, the studies after the regulation point basically were saying, well, there's not really this major beneficial effect from statins because the industry-supported scientists couldn't continue to do what they were doing. So the benefits that we saw from all of this clinical research before they put in the regulations, well, it's very questionable at this point. So if you're going to do a meta-analysis to get a determination on, are statins worthwhile to use, then you probably wouldn't want to include data before 2004, which is what these researchers are basically arguing for. So if you have meta-analysis that's looking at studies prior to 2004, then it's possible that it was influenced by industry-supported scientists or by the pharmaceutical industry in and of itself. And I'm not going to go over this one quite as in depth, this chart, but this figure, or figure 1 here, goes directly with what they're saying. So here they have studies after 2004 and studies before 2004. They have coronary artery events as a percentage, so you can see if they're increasing or decreasing. And then you have LDL-C levels. So they're plotting LDL-C levels against coronary events. And these are the studies that were done after 2004 and the ones that are due before. And what essentially they're finding is after the 2004 regulations, the effect was either quite small, there was really no effect, or the statins may have possibly made things worse. And they talk about different diseases: diabetes type 2, coronary artery stenosis, heart failure dialysis, high CRP, which is C-reactive protein, which is an acute phase inflammatory protein that is associated strongly with heart disease, or high-risk groups. I don't know how they define those high-risk groups. So you can see the difference here. They plotted it, and there's a little blurb about it here. So if you get a chance, if you ever pick up the study, you can look at some of this. There's quite a few awesome graphs in here. So the next piece they get into is statins are mitochondrion toxic. So again, for anyone who's unaware, mitochondria are the engines of the cell. And what happens is the statins impair their function, their ability to produce ATP, or AKA, depending on which theory you're looking under, ATP is considered the energy of the cell, although if you get into any of Gilbert Ling's stuff, you may come out with a different perspective, but that's a different story. And so what they go into here, and I'm going to read this here, is they say CoQ10, both in its oxidized ubiquinone and reduced ubiquinol forms, and heme A are essential components of the electron transport chain and are synthesized from prenal intermediates in the cholesterol biosynthetic pathway. Statins inhibit CoQ10 and heme A biosynthesis and thereby ATP generation. ATP is essential for normal heart muscle function, metabolism of cellular components, and other activities in cell life. Cholesterol is a major component of cell membranes, functioning to maintain their integrity, which is likely to be affected by statins. Thus, statins are mitochondrial toxins, making all cells ATP depleted because most mammalian cells depend on mitochondria for their energy metabolism. Statins are general cell toxins. And so what they come into over here on this graph, and this will kind of explain it. So CoQ10 is this little shuttle that shuttles some of the electrons from complex 1 and complex 2 over to complex 3. As this goes through, you pump out the hydrogen ions, and that creates a gradient that will, down here, those hydrogen ions flow back through, kind of like a water wheel, and drive this little pump, or it's not really a pump, but it's an energy generator. It's kind of like a hydroelectric energy generator called ATP synthase. Now, complex 1 is where NADH is oxidized to NAD+, and then complex 2 is where FADH moves into and is oxidized to FAD. So fatty acids generate larger amounts of FADH2, and carbohydrates generate larger amounts of NADH. And this is through beta oxidation, and then with carbohydrates, it's at the through the Krebs cycle, or glycolytic or oxidative phosphorylation. So CoQ10 is very important in making sure that the electron donors or carriers that are generated by carbohydrate oxidation and fat oxidation are oxidized, and then their electrons can be passed along to complex 3. Now, CoQ10 is completely dependent upon prenyl intermediates in the cholesterol biosynthetic pathway for its production. If you don't have adequate prenyl intermediates, you cannot produce CoQ10. So statins, by blocking the cholesterol biosynthetic pathway, which includes these prenyl intermediates, leads to a decreased production of CoQ10, which over time will, because there's going to be a turnover of CoQ10 in the mitochondria, will directly impair mitochondrial function. This is not good, especially considering that in the diabetic or the heart disease or the congestive heart failure states, the energy production of the cells are already taxed. So imagine making it worse through multiple mechanisms. Second piece, Heme A is a certain type of iron-containing protein, and it's important in cytochrome C oxidase structure. Heme A is dependent on those prenyl intermediates as well. So a lack of prenyl intermediates due to the statin use blocking their production can also impair the Heme A cytochrome C complex. That is very, very bad in conjunction with blocking CoQ10. So you're blocking cell respiration at 2 steps. In an organ like the heart, which has a high amount of energy production and utilization, this is like a terrible, terrible thing overall, or a possibly terrible thing overall, because you're basically impairing its ability to use both carbohydrates and fats to produce energy. And usually heart failure is a progressive loss of the heart muscle's ability to adequately produce energy and to adequately circulate the blood supply. So the heart starts to essentially become weak. That hypertrophy that you see is not necessarily because the heart is becoming stronger, but more so because the heart muscle is trying to compensate for a decreased pumping ability or having to pick up slack for other areas of the heart that are either stiff because they're unable to relax, or because they're not pumping effectively, or because the vasculature throughout the rest of the body has a high amount of resistance. So there's multiple factors going on. It essentially boils down to issues with energy metabolism, whether directly at the heart muscle or with inflammation and damage in energy metabolism throughout the rest of the vasculature. The ability for the vasculature to relax throughout the rest of the body is dependent upon the energy state of those cells, as well as, which includes exposure to some of the stress hormones, catecholamines, the glucocorticoids, cortisol, aldosterone, and then also the renin-angiotensin products like angiotensin 2 and then endothelin 1, etc. So there's a lot of this is dependent upon what is the actual energy state of these cells overall, and also the general energy state of the body and the exposure to some of these stress pathways. So impairing the mitochondrial function at the heart or at these other cells by depleting CoQ10 or Heme A with a statin can actually make the situation, or possibly theoretically, or perhaps in practice, as these authors suggest, significantly worse. So it's definitely not a good idea to use a statin to lower cholesterol, but in that process also lower CoQ10 and Heme A and impair energy production even further. So what the authors continue to say here is CoQ10 is an essential cofactor in electron and proton transport and mitochondrial energy production, as well as in several other aspects of cellular metabolism. The bioenergetic effect of CoQ10 is believed to be of fundamental importance in its clinical application, particularly as it relates to cells with exceedingly high metabolic demands, such as cardiac myocytes. The reduced form of CoQ10, ubiquinol, is recognized to be a clinically relevant antioxidant in different cellular compartments, especially the mitochondrial membranes, where it protects mitochondrial DNA from damage. It is well known that mitochondrial DNA is much more vulnerable to oxidative damage than nuclear DNA. So besides functioning in energy metabolism, which they do discuss here in CoQ10, and also just an off semi-tangentially related concept here, but one of the best sources that's discussed inside the ancestral community of CoQ10 is generally heart. And the reason why heart is, if you look at the physiologic levels, the heart is, you essentially have a muscle that's contracting hundreds of thousands of times a day. I don't know the specific number offhand, but so I guess say you have 60 beats per minute, and then you're contracting 60 minutes in your hour. So there's 6 times 6, so 300 or 3,600 different beats just there alone. So that's per hour, and then you do that times 24 of those, and you're at 86,400 beats in a day. So 60 beats per minute by 60 minutes in an hour by 24 hours in a day, it's 86,400 beats. So you have a muscle that is working pretty significantly. And again, 60 beats per minute is actually technically on the lower side. That doesn't incorporate any type of movement or exercise, which can increase heart rate, or people who have higher resting heart rates. So you have a lot more, you have a muscle that's working quite hard. So it's going to go through a decent amount of energy in the form of fatty acids and carbohydrates. And in order to do so, it's going to require large amounts of CoQ10 in order to shuttle the electrons through the different enzymes within the electron transport chain. So depleting that of the heart is, quite problematic. So I just covered that. They also mention it here. The other thing that they talk about is when you deplete CoQ10 inside the mitochondrial membrane, which the mitochondrial membrane is one of the main areas exposed to ROS, because this is where you are producing large amounts of energy. You essentially have a slow combustion process involving the transfer of electrons to different proteins and components, and having oxygen inside the mitochondria as a final electron acceptor. So you have quite a bit of possible areas where reactive oxygen species can be produced, and then those reactive oxygen species can then damage the membrane. And also the mitochondria has its own set of DNA, and that DNA is very liable to be damaged by exposure to reactive oxygen species. And doing so, as I'll talk about, or Jay and I discussed on one of our research study episodes that we did, the mitochondrial DNA can handle a certain amount of disruption or mutation to some extent before the mitochondria stop functioning. But at a certain threshold, it directly impairs mitochondrial replication, the formation of new mitochondria, and the function of mitochondria, because that DNA transcribes or leads to the transcription and then translation of different proteins that are necessary for mitochondrial functioning. So having a large amount of ROS exposure without protection from things like CoQ10 or some of the antioxidant enzymes, superoxide dismutase, glutathione peroxidase, etc., without having these things present, and you can start to damage the membrane, and then you can of the mitochondria, and then you can also start to damage the mitochondrial DNA. And over time, you degrade that structure of the engine long enough, and it'll start to basically not function. The difference between the human engine or the mitochondria and like a car engine is that the human engine is self-repairing given the right necessary resources, and then it can also, these little mitochondria can undergo like a breakdown or like an apoptosis to some extent, or like a mitophagy, which is essentially and part of the mitochondria gets digested or adjusted if it's broken or whatnot, or it's essentially like a house cleaning process, and then they can be rebuilt appropriately. So if you give the right resource, you can do those things. However, there is obviously a threshold for that damage. So chronically running low CoQ10. Also, another thing that's discussed is using high linoleic acid oils to lower cholesterol. Combining that with statins is probably an extremely terrible idea if you start to load up this mitochondrial membrane with polyunsaturated fatty acids, and then you deplete its ability to produce energy, and you cause backlogs at complex 1 and complex 2 with subsequent large amounts of ROS generation coming from these complexes, then interacting with the omega-6 or the omega-6 polyunsaturated fatty acids at the membrane is just a massive recipe for mitochondrial failure, and then subsequent getting stuck in glycolysis and also like cellular failure, probably like destruction of the cell in and of itself, hopefully through apoptosis, but possibly through necrosis and other means. So ideally, you don't want to be incorporating large amounts of oxidative substrate or components that can be oxidized easily in the membrane, like polyunsaturated fatty acids, and then also impairing energy metabolism directly, and also depleting antioxidants inside the mitochondria as well. It's literally like a recipe for absolute disaster. So as the authors continue here, they go on to say, and here's the title of the next section: decreased ATP generation and resulting cell damage contribute to the development of coronary heart disease in familial hypercholesteremia cases and in statin-treated people. So what they say here is the initial pathology or pathophysiology of the onset of atherosclerosis has not been well defined. So we don't 100% know all the mechanisms of what is initially causing this atherosclerosis, although it's probably multifactorial, not just because, cholesterol. But they say, however, any tissue damage, whether derived from a pathogen or non-infectious damage, so like shear stress from like in the high pressure areas of the heart, or from a pathogen or, for example, endotoxin, may induce inflammation to repair damaged tissues, leading to many diseases, including atherosclerosis. So exposure to pathogens, bacteria, parasites, fungi, microbes in general that are looking to gain access to the nutrient-rich reservoir of the human body in the form of blood or other components, or even their components of their cell walls, such as what you see with endotoxin, or even other components that aren't cell walls, but other proteins that are part of their structure. When the immune system gets a hold on those, or if you have damage to the vasculature, say you have vitamin C deficiency and you have shear stress from the large amounts of pressure from blood flowing through the vessels, you can induce an inflammatory process. That inflammatory process, to some extent, at least described here by the authors, is initiated to repair damaged tissue. So the immune system moves in, cleans up the pathogenic components, and ideally can help to repair those damaged tissues. Now, this may lead to diseases if this repair process, again, if you don't have the right resources, or you have an overwhelming amount of damage, overwhelming the system's ability to repair, then you can get, essentially crappy repairs or impaired repairs, and that can lead to things like atherosclerosis. And in atherosclerosis, as a side note, what you tend to see is macrophages, which are essentially white blood cells that can gobble things up. Think of like they're little Pac-Mans, I guess is a good idea to understand them. They can come in and they can gobble up that cholesterol that's inside the bloodstream, so they essentially gobble up oxidized cholesterol, and I'll go through another study discussing this, and then they pull it across the vascular space, and then the oxidized cholesterol it protects the endothelial cells lining the vasculature, but what can wind up happening is those macrophages inside with these oxidized cholesterol and perhaps other polyunsaturated fats can start to become damaged themselves, and then they build up, become bloated. They're called foam cells at this point, and they're actually in the muscular layer of the arterial wall, protecting those endothelial cells lining the lumen of the vasculature. And then over time, those foam cells can die, and you can get necrotic pieces and an unstable plaque. That plaque can break off, and that's where you see the occluded vessels or the embolisms, the emboli that form. The thrombus is the actual plaque that forms on the wall. I know it's a bit of a tangent, but what they continue to say is these inflammatory repair mechanisms are mediated through toll-like receptors in response to activators produced by infections, hypoxic ischemia damage, overwork, and/or stress and elevated advanced glycation end products. The associated coronary artery stenosis leads to decreased blood flow and reduced supply of nutrients and oxygen, leading to decreased ATP generation in blood vessels and heart muscle cells. So you have this inflammatory stimulus like I was describing. The immune system gets activated. The toll-like receptors are the receptors on the immune cells that recognize these different either damaged cells or viral proteins or fungal or bacterial proteins. They get activated. That stimulates a whole entire immune cascade that there's multiple effects that go from there. And that can lead to damage if the actual infection can lead to damage, or the damaged tissues are already damaged. The immune system comes in. The chronic elevation and inflammation overall can exacerbate that damage. You can also have a hypoxic ischemic damage, so where a lack of blood flow to somewhere can lead to an ischemia or a lack of oxygen. and that or a hypoxia, so a lack of oxygen, so a decreased supply, and then that causes those tissues in that area to become damaged. There also can lead to overwork and or stress. So basically putting an excess energetic demand on tissues that have a limited energetic supply. There's a limit to the amount of energy we can produce. That can cause damage to those tissues as well. And an elevated advanced glycation end products, which inside the vasculature are usually more associated with issues with metabolism and or excess PUFA consumption, rather than just you had too much fructose. The fructose consumption that you have generally doesn't lead to high levels of serum fructose levels. It's usually metabolized in the liver before any fructose or much of any fructose reaches circulation. So this is more of a PUFA and issues with glycolysis or so like a metabolic issue than just you have too much carbohydrates. So this damage inside the vasculature can then lead to a narrowing of the vessels in arterial stenosis that further decreases blood flow and reduces supply of nutrient oxygen to the downstream tissues, which without glucose and oxygen or fatty acids, you get decreased ATP generation in the blood vessels and heart muscle cells. So the areas that get occluded can produce less energy because they don't have the resources to produce that energy. So it becomes this kind of vicious cycle. They have a little tangent here about familial hypercholesterolemia, but I'm not going to go too in depth in it. But essentially what they're talking about here is, in the case of familial hypercholesterolemia, supply of nutrients, particular fats to peripheral tissues, is restricted from early age due to defective or deficient LDL receptors. So hypercholesterolemia means high cholesterol in the blood. It is a genetic disorder. The reason that the hypothesis why this happens, or the theory that's proposed as to what goes on, is that they're LDL receptors. The receptors for the proteins that carry lipids from the liver to the tissues doesn't necessarily work. So their bodies circulate higher amounts of the of LDL. So they call it hypercholesterolemia because they generally have very high cholesterol levels in the blood. And it's not because they're overproducing usually, it's more because their tissues aren't taking up the lipids from the LDL, which also, according to these authors, leads to a deficiency of lipids at those tissues since they are not able to effectively pull them out of circulation because of the defective receptor. So they're talking about it as that leading to like an energy deficit in those types of people. They go on to discuss that this leads to decreased ATP generation and cellular damage. Walter Hartenbach, former professor of pathology at München University, observed cellular damage in the artery well before fatty acid plaques were formed. So their argument here is that the decreased ATP generation and cellular damage occur from a lack of functioning LDL receptor that then predisposes towards the fatty plaques, or they are implying that it predisposes towards the fatty plaques, rather than the fatty plaques being just because they have too much cholesterol circulating in their bloodstream, which that they're providing different mechanisms besides this, high cholesterol causes heart disease piece. They're actually trying to look into more what are the nuances, and they're coming at it from a bioenergetic perspective, not because they necessarily follow Ray Peat, but this discussion of energy production is in the research called or termed bioenergetics. I don't know how much milk or orange juice, and I'm using that as a cliche here, that the researchers have on a regular basis. So as they continue, they say, in the case of statins, ATP generation is impaired by their inhibition of CoQ10 and heme A biosynthesis. Similar to the case of coronary heart disease and familial hypercholesterolemia, limited supply of ATP could be a major cause for heart muscle and coronary artery damage. So basically they're just wrapping up what I explained with multiple tangents. That lack of ATP generation due to statins blocking the necessary components to build CoQ10 and heme A, with CoQ10 and heme A being extremely important for energy production, can lead to heart muscle and coronary artery damage because now these tissues have depleted energy, or depleted ATP, which is extremely necessary and beneficial to their function. Then they have here some presumed factors leading to atherogenesis. This is kind of an overview of what they've talked about so far. So over here they have coronary heart disease. So conditional infections, persistent inflammation, advanced glycation end products, overworking stress. Those are some of the things they mentioned in the previous paragraph. Those lead to stenosis of the coronary arteries through the inflammation and buildup of plaques in the arteries. And then you have a restricted supply of energy and oxygen. So decreasing the lumen size, essentially the amount of space that the blood has to move through, restricts the supply of energy and oxygen to tissues downstream of that blocked lumen area. And then that can lead to decreased ATP generation. Next piece is in the familial hypercholesterolemia. There's impaired LDL receptor function, so they can't take up the lipids from the LDL carrier lipoprotein. So that decreases the amount of energy that these people actually are able to produce because they have decreased substrate going to those cells. That decreases ATP generation. Statin administration decreases the prenal intermediates. So the prenal intermediates are some of the intermediates in the pathway that produces cholesterol. That impairs the production of Heme A, CoQ10, selenoproteins, and vitamin K. So we'll cover these next. The lack of Heme A and CoQ10 directly decreases ATP generation, as we just discussed. The decreased selenoproteins decreases the peroxidative enzymes, which increases peroxidative stress. The lack of vitamin K doesn't allow for adequate function of the matrix GLA proteins, which function to help protect against arterial calcification. So then that leads to arterial calcification. And then the combination of arterial calcification, excess or oxidative damage, and decreased ATP generation leads to impairment of muscle cells and onset of heart failure and atherogenesis. So they're bringing together a multifactorial approach rather than just saying, oh, look, high cholesterol causes heart disease. There's multiple factors going on here centering around decreased ATP generation, but also involving arterial calcification, oxidative stress, and a restricted supply of energy and oxygen due to decreased lumen size of the vasculature. So this is a much more complete theory. Is it 100% correct? I mean, that's yet to be vindicated and validated, but there's definitely quite a few factors on here that make a lot of sense and are in line with a lot of the research that's coming out. And it makes sense because these researchers are pulling from the research to try to figure out what's actually going on in heart disease instead of just saying, oh, everyone just needs a statin. Or when I was in high school, I actually sat in some medical school classes. It was a program offered in the high school. And I remember hearing one of the doctors who was a professor there was saying we should just put statins in the water supply. And even at that point in high school, I was reading Dr. Mercola's blog. I was getting into the intermittent fasting and low carb and keto and paleo, and I was very involved in all that stuff. So I'd already gone through the basically realizing that the LDL or the cholesterol hypothesis of heart disease didn't make much sense. So I remember coming home and just having an absolute fit about what the doctor was saying, just how ridiculous it was, and they don't know what they're talking about and all this type of stuff. I don't have those fits, at least publicly or outside of my own mind anymore. But I remember hearing that. So they're promoting a better hypothesis overall. As they continue, they start to get into here to statin administration and selenium deficiency causes heart failure through a common mechanism. So that's what I was talking about before with at least initially in the preface, was that they can work. So a selenium deficiency and a statin administration can cause a decrease in selenoproteins through different mechanisms. And what you see in selenium deficiency, which is called Keshan disease, you often see heart failure. So if statins are also decreasing the same selenoproteins, even though it's through a different mechanism, you could assume that you would probably get the same result because what's causing the heart failure is the lack of these proteins. So we'll jump into it. What they say is selenium is an essential trace element and it is incorporated into selenoproteins using a transfer RNA that is specific to the selenocysteine tRNA. A minor base of the tRNA, isopentenyl adenine or adenine, is synthesized from a prenyl intermediate, and its synthesis is inhibited by statins. So again, statins inhibit prenyl intermediates. This messenger or this transfer RNA requires one of these prenyl intermediates to be created, and the transfer RNA is very necessary for the selenoproteins. So if you don't have the prenyl intermediate, then you don't have the tRNA, then you don't have the selenoproteins. They say here, and this is where the Keshan disease thing comes from. In the Keshan province of China, dilated cardiomyopathy was common, which was later revealed to be due to selenium deficiency. They're setting the preface here. So selenoproteins include glutathione peroxidase, iodothyronine deiodinase, thyroreductin reductases, and more than 10 other kinds of selenoproteins. When glutathione peroxidase synthesis is inhibited by statins, peroxidative stress is elevated, which is generally accepted as causative for atherogenesis, carcinogenesis, and aging. Statins also lower the levels of anti-peroxidase enzymes such as superoxide dismutase and catalase by unknown mechanisms. So these are important selenoproteins. Glutathione peroxidase they talk about here is involved in protecting against oxidative stress. Iodothyronine deiodinase is another actually important enzyme that takes an iodine group off of T4, which is thyroxine, to create triiodothyronine, which is T3, which is active thyroid hormone. So if anyone's familiar with Broda Barnes' work, active thyroid hormone is involved, or T3 is heavily involved in taking cholesterol, transporting it into the mitochondria of the cell. So this is a combination of Dr. Barnes and Dr. Pete's work. And so Danny has also discussed this heavily, Danny Roddy. But essentially, it takes the cholesterol and it brings it inside the mitochondria. I think that's through the STAR protein. And what winds up happening is that cholesterol is converted into steroid hormones. So in Dr. Broda Barnes' work, giving his patients natural desiccated thyroid, the work that he did, like looking at rates of heart disease, heart attacks, etc., the patients of his that received the natural desiccated thyroid hormone therapy and achieved the parameters that he set for the body temperature and whatnot, led to significantly decreased heart attacks and heart disease in that population. And so they don't discuss this directly in the article, but impairing the creation of active thyroid hormone by decreasing the deiodinase enzymes could actually be another significant mechanisms by which statins directly or worsen heart disease for people. So something interesting to keep in mind. They further on discuss that reactive oxygen species, or oxidative peroxidative stress, they call it here, is accepted as a causative agent for atherogenesis, carcinogenesis, and aging. It is indeed. That's even in the mainstream. That's pretty much a known fact at this point, or it's a known fact that the reactive or the production or excess production of oxidative stress can lead or produce these issues by damaging the cellular components and structures, especially the mitochondria. The last thing that's extremely important is that superoxide dismutase and catalase, both enzymes that protect against oxidative stress, also seem to be lowered by statins. So you're lowering their glutathione enzymes, superoxide dismutase, and catalase. You're directly blocking three very potent enzymes that inhibit oxidative stress or mop up reactive oxygen species. So that's a pretty big factor. That's actually a huge problem as well. Especially if you're going to impair energy metabolism up here by blocking CoQ10 production in Heme A, you will get more ROS through these complexes. You will get, because you'll have a buildup of NADH, you'll have a buildup of FADH2, but you won't be able to transfer the electrons appropriately through the CoQ10 carrier. So then you can have a buildup, and you see this in partly in the Randall cycle and fatty acid ROS generation, is when CoQ10 doesn't move to complex 1 because there's excess FADH2 at complex 2 kind of hogs, I guess is an easy way to think about it, but hogs CoQ10, and you get increased ROS at complex 1. With all of that happening, if you're blocking CoQ10 altogether and Heme A, then you're going to start producing more ROS here, and then CoQ10 is an antioxidant. The glutathione enzymes are important in antioxidant defense. Superoxide dismutase and catalase are also extremely important. So you're just basically defeating the cell. It's like, I can't produce energy, and I have all this ROS, and I can't even protect myself from that ROS. It's just a recipe for disaster on a metabolic level. Researchers continue. They say, in accordance with the mechanisms described above, glutathione peroxidase activity in erythrocytes was shown clinically to be inversely associated with coronary heart disease events and positively with event-free survival when patients with coronary heart disease were followed up for 5.4 years. So that's figure 6. Let's see where we're at. So here's figure 6. Essentially what you're seeing is erythrocyte peroxidase 1 activity, units per gram of hemoglobin, and then you're seeing hazard ratio for coronary artery disease events, and then hazard ratio for event-free survival. So the coronary artery disease events is blue. The event-free survival is red. So essentially what you want to see, so we'll start with the blue. What you're seeing here is that with increased, and down here on this is the units per gram of hemoglobin of the red blood cell peroxidase 1 activity. So obviously the higher this number over here, the higher the peroxidase activity. So what you're finding is that the coronary artery disease events have an inverse association. So the more peroxidase 1 activity the red blood cells have, the less coronary artery disease events are present. And then the more erythrocyte peroxidase, so red blood cell peroxidase activity that you have, the greater the event-free survival. So the less heart disease events you have, and with the greater survival. So you're seeing this, you have an inverse trend with coronary artery disease events, and then you have a linear positive trend with event-free survival. So blocking glutathione peroxidase with statins by inhibiting the production of those prenatal intermediates that are necessary for the tRNA and then for the production of the selenocysteine proteins can impair the production, can basically lead to increased, or conceptually should lead to increased coronary artery disease events by lowering the erythrocyte peroxidase activity, and also decrease the event-free survival by giving us lower erythrocyte peroxidase 1 activity. So just an interesting, they're putting the pieces together here, and it's actually done pretty well overall. And they're looking at the association, and they're looking at mechanisms and trying to put together a picture. Other thing I want to talk about here is this graph here. They say comparison of skeletal muscle properties in the leg between statin users and statin non-users. So they have a ratio here, statin group in ratio to control group. So the statin group on top, control group on the bottom. So what you're seeing here is that the statin group's systolic blood pressure tended to be higher. Their hemoglobin A1C tended to be higher. Their glucose area under the curve tended to be higher, and their insulin area under the curve tended to be higher. So the insulin area under the curve, it says NS. That wasn't significant, but all of the other ones with stars are significant. So a p-value of less than 0.05, meaning that there's a 95% chance that this wasn't due to chance. Now, what you're seeing is that the statin group had lower CoQ10, lower catalase, which is an antioxidant enzyme, lower manganese superoxide dismutase, which is an antioxidant enzyme, decreased glutathione peroxidase, which is an antioxidant enzyme, decreased uncoupling, decreased complex 4 activity, decreased myosin heavy chain, which is a component of the muscle protein structure, decreased oxidative phosphorylation, and then again a decreased oxidative phosphorylation here. They look for a specific, concentration of glucose. So overall, you're seeing worsened blood pressure, glycation, blood glucose levels, and a non-significant increase in insulin, and then decreased antioxidant defenses and mitochondrial function. So overall, it's really not a good picture for statin use, at least based on this. And they say here in the chart, as compared with non-users, statin users exhibited higher systolic blood pressure and elevated glycated hemoglobin levels of CoQ10, anti-peroxidative enzymes, uncoupling proteins, complex 4 of the mitochondria, and myosin were lower, oxidative phosphorylation ability was lower, and glucose levels and glucose tolerance test was higher. So again, everything I just basically discussed, just not a good profile overall metabolically. Next piece here is the next thing that they discuss. I know this is quite an intensive article, but statins inhibit vitamin K2 synthesis and accelerate artery calcification. So this is where they get into the matrix GLA proteins and vascular calcification. So what they say is the enzyme synthesizing vitamin K2 from vitamin K1 are present in many tissues, including the brain, and statins inhibit the conversion of vitamin K3 to vitamin K2 by restricting the supply of isoprenyl intermediate. Vitamin Ks serve as cofactors for an enzyme catalyzing gamma carboxylation of glutamyl residues in proteins such as coagulation factors, osteocalcin, and matrix GLA proteins. Vitamin K serves as the cofactor in the carboxylation of matrix GLA protein present in bone, blood vessel, lung, heart, and kidney soft tissues. In the gamma carboxylated form, the matrix GLA protein retains capacity to bind calcium and protect blood vessels from calcification. Statins inhibit vitamin K2 formation and thereby accelerate coronary artery calcification, an important marker of the progress of atherosclerosis. So I'm going to turn this into English because like all of these terms are, they're very much in-depth research terms. It's the research lingo. But it's an important concept. And then before I get into that, what I do want to discuss is there's been quite a bit of research as far as looking into what is some of the best tests in determining heart disease. And coronary artery calcification has been determined to be one of the best tests to determine progression of heart disease. I will go over some studies in the future discussing coronary artery calcification, but progression of coronary artery calcification over time is one of the biggest things that research is starting to look to as far as determining heart disease besides just total cholesterol and whatnot, because their associations there have only been associations and haven't yielded as much value as they had hoped. So tangent aside, what they're discussing here is that vitamin K2 is a cofactor for a particular enzyme that adds these special groups onto these important biological components. So you need basically what you can think of is vitamin K flips the switch on these, or vitamin K in conjunction with this specific enzyme flips the switch on these very important proteins or factors inside the body. So if you don't have vitamin K flip that switch with the other enzyme, then these coagulation factors, osteocalcin, and matrix GLA proteins don't work. Why is that so important? Because the matrix, so for a couple things, you need coagulation factors. You don't bleed out if you get a bruise or you fall or you cut yourself, so it's extremely important. Next piece is that osteocalcin is extremely important component in the development of bone. So vitamin K is extremely important in the development of bone, making sure that the calcium that you ingest and that your calcium supply is moved towards your bone production rather than calcification of your tissues. And then matrix GLA protein is an extremely important protein in protecting the vasculature, your blood vessels, from being calcified. So as I was just discussing, coronary artery calcium score is a great indicator of heart disease, or it's coming out to be so in the research. And what is protecting against this calcification is the matrix GLA protein. And what does the matrix GLA protein need to work? It needs adequate amounts of vitamin K2. So not having adequate vitamin K2 basically impairs all of these processes: bone formation, coagulation factors, and then protecting against calcification of soft tissues. Now, statins block the formation of vitamin K2 because the isoprenyl intermediates that are involved in the production of cholesterol are necessary components for the formation of vitamin K2 from vitamin K1 or vitamin K3. So you really need to have these isoprenyl intermediates, and statins basically are blocking this whole process. And so here's another mechanism by which now not only is there increased or mitochondrial dysfunction, there's increased atherosclerosis, there's increased reactive oxygen species, and then on top of that now you have an increased risk of calcification through blocking all these pathways. So do statins lower cholesterol? Sure. But does that solve the problem? No. Now what we're saying is maybe it even makes things way worse. So you want to, if you're going to take a drug, what you want to do is, or a compound or a food or whatever it is, you want a risk-benefit analysis that's, I have a lot of benefit with very minimal risk. So at the minimum, you want to be doing something that's not harming you. At the maximum, you want to be doing something that is not only not harming you, but providing you with a significant benefit with very minimal downside. Statins are at best not really helping, and then at worst actually making things significantly worse. So, or possibly making things significantly worse through these different mechanisms. So it's just, they're building a very good case here against statins. So they continue here with the vitamin K piece. They say when vitamin Ks are used as cofactors, they are reactivated in tissues. Therefore, vitamin K deficiency is generally considered uncommon, except for the case of long-term administration of warfarin as an anticoagulant. Chronic administration of warfarin is known to accelerate artery calcification. Although not directly related to statins, dihydrovitamin K1 produced during partial hydrogenation of vegetable oils is not converted to vitamin K2, and its administration leads to tissue vitamin K2 deficiency, which might be associated with atherogenesis. So, couple things. Why are they bringing this up, first of all? So, if you're depleting vitamin K, in cases where vitamin K becomes depleted, such as seen with hydrogenated vegetable oils, and then also with warfarin administration, you see vascular calcification, arterial calcification. So you know when you don't have adequate vitamin K, or you can't recycle the vitamin K that you have, or produce the proper functioning vitamin K, you get arterial calcification. So we know that statins decrease vitamin K by blocking the isoprenyl intermediates, and we also have evidence that these other substances that can block that block vitamin K lead to arterial calcification. So essentially, it would make sense that statins would also do the same. And obviously, warfarin is another drug that's commonly used as an anticoagulant, and it does cause vascular calcification. That is known, which obviously there's another problem there for a supposedly a drug that's supposedly helping heart disease. And then again, vegetable oils are supposedly helping heart disease as well, although the hydrogenation thing has come under fire recently, so that's pretty good overall. But the vegetable oils have been known to cause problems, and or have thought to be good for heart disease, and now what we're finding is that they cause significant problems. And here's another possible mechanism for hydrogenated ones, specifically by leading to production of dihydrovitamin K1 that cannot be converted effectively to vitamin K2, which can further lead to a vitamin K deficiency. As I continue, in a clinical study of diabetics, high-frequency statin users were shown to exhibit accelerated coronary artery calcification compared with low-frequency statin users. Incredibly, the lead author chose to interpret this increase in coronary calcification in a positive light by speculating that statins may lower the lipid-rich core of atherosclerotic plaques and may enhance the density of calcification as part of the healing process, potentially contributing to plaque stabilization and decreased cardiovascular disease events. So, interestingly, in diabetics, higher frequency statin users had increased coronary artery calcification. So this, to some extent, is vindicating these mechanisms. And then furthermore, which I find quite interesting, is the lead author on the study tried to interpret that effect of the statins as a positive thing. So it's like, well, statins must be good. So how can this be framed as a positive mechanism? And basically what they're saying is, since statins decrease cholesterol, well, maybe the lipid-rich core of those plaques was decreased, so the amount of lipids in your plaque is decreased, which increases the density of calcification. So since there's less lipids, there's probably more calcification as part of the healing process. And then the increased calcium, well, that must just stabilize the plaque and decrease an embolus formation, which is ridiculous because calcification is known to be the end stage of the inflammatory process, especially with fibrosis. So atherosclerosis is literally arterial hardening, and calcification is like the end stage of that. So increasing that process in other situations would actually be considered not a good thing. However, here, because it's with statins, well, it must be a good thing, just because statins are supposed to be good. So it's a very, you have to be careful with interpretations of research. You have to really look into what's going on. I mean, when you look through the studies, you have research, like even here, this is a researcher's interpretation. So it's important to also look into the mechanisms of the studies. Like when you look into the caloric restriction studies, that I had previously gone over, or in the other YouTube videos that I have, you can see that I discuss, I go through like what exactly did the researchers do, so that we're not just relying on their interpretation. Although it's not that it's always wrong, it's just important to look at the work in general, because you can get some weird interpretations like this. Next piece here, they say Nakazato et al. Evaluated coronary computed tomography angiography in 2,413 patients on statins and 4,260 patients non-statins. None of the subjects had any known coronary artery disease. Statin use was associated with a significant increase in the prevalence and extent of coronary artery plaques containing calcium. So the coronary artery computed tomography angiography is that's how they determine the calcium score. And essentially they're taking CT scans, which it does involve radiation, of the blood vessels, the coronary blood vessels, and looking what the CT scan can pick up is the density of the increased calcium. So you can get a score. There's a certain basically calculation you can do with that to get a coronary artery calcium score and determine the amount of calcium inside those plaques. So what they're showing here is that the statin users had an increased, or there's an association of statin use with increased amount of coronary plaques containing larger amounts of calcium. Next piece here, next title here, clinical trials showing or suggesting that statins increased atherosclerotic disease and heart failure. What they talk about here is there's a Japanese study. This was the first, they go on to say, this was the first large-scale intervention trial with a statin performed in Japan, and those with total cholesterol levels greater than 220 milligrams per deciliter were treated with a low-dose simvastatin for 6 years with no control group. So they didn't have a control group. It's not ideal, but the horizontal axis in figure 8 is plotted with total cholesterol levels after treatment, the mortality rates for cardiovascular disease, cerebrovascular disease, which is strokes, or TIAs, transient ischemic attacks, cancer, and all causes were elevated along with decreasing total cholesterol levels from 220 milligrams per deciliter. So they go on to say the higher mortality rates in the higher, or I'm going to skip this actually because this goes into another piece. But I'm going to go to the graph really quick and show you guys what they're talking about. So here's total cholesterol on the bottom after simvastatin, so it's in milligrams per deciliter. Here's relative risk or mortality. So what they find is actually, ideally what you would see is as you decrease cholesterol, mortality from all causes, cancer, stroke, and cardiovascular disease should decrease. But what you're seeing is that at low cholesterol levels, really low cholesterol levels, you're seeing an increase in mortality, cancer, stroke, and cardiovascular disease. Smaller amounts down here. And then then in the 180, 220, 200, 240, it seems that there's not really, things are they're at the lowest. They're at the bottom of the U. And then at extremely high cholesterol levels, you also see this increase of risk of mortality. So there could be other things going on here that could be in discussion, but essentially what you're seeing is that now this is very important because the recommendations are to have cholesterol less than that 200 in these older people with the cardiovascular disease. So you're at this again. This is an association study, but I've seen this curve discussed before in other blogs, and this is not ideal. You do not want to see, okay, I'm giving people a statin to lower their cholesterol below 200, but their all-cause mortality, their cancer, their stroke, and cardiovascular disease risk is either not doing much or getting worse. So it's why, especially with the other known side effects of statins, which are known from the modern medical perspective, like the problems with myopathies and whatnot. With that known, if it's not doing anything better, there's no need to take that drug. So just an interesting association study to point out there. We're going to skip past some of this stuff. It gets a little bit, it's like a reiteration of a lot of these things. So what we go to here, we're going to go down here and go to the clinical impact of statin-induced depletion and supplementation of CoQ10, statin-induced CoQ10 depletion, and muscle damage. So what they go on to discuss here is that statins' adverse effects on skeletal muscle are the most commonly reported statin side effects. Skeletal muscle weakness, muscle pain, and skeletal muscle death with elevated creatinine kinase levels are a well-recognized phenomenon among prescribing physicians and patients alike. This is known, even working in the hospital as a nurse, this is a known side effect of statins. One of the places I actually worked at, one of the hospitals, actually one of the doctors really liked giving their patients CoQ10 with their statins because of some of these effects. So what they go on to say here is statins have been demonstrated to decrease the concentration of mitochondria in muscle, oxidative phosphorylation capacity, and the skeletal muscle mitochondrial DNA levels. So all not good things. You're decreasing the engines of the cells, you're decreasing the ability of the cells to produce energy, and then you're decreasing the ability of those engines to reproduce themselves inside the muscle cell. So you're depleting multiple levels of energy production in the muscle cell. In view of this obvious skeletal muscle toxicity, it would be naive to assume that statins would not likewise negatively impact the much harder working heart muscle cells, which have exceedingly high ATP requirements. Animal data, indeed, in animal data, statins have been shown to increase mortality in cardiomyopathic hamsters and increase ischemia reperfusion, heart muscle damage in dogs. So, what they're basically showing is that if we know that statins damage regular muscle tissue, and the heart is muscle tissue, and it's muscle tissue that's working significantly harder than the rest of the muscles. It has to work 24/7, 365. There's no sitting on the floor so that you can relax your quads for your heart. Your heart is always going. If it stops, it's game over. What they're basically saying is that we know that statins damage regular muscle, so they would be naive to assume that they also wouldn't damage the heart, which is a muscle that's working even harder. And then basically they cite some studies to show that they do in at least in hamsters and dogs worsen heart function. And so that leads them into discussing here evidence for a causative role for statins in human heart failure. So the first reported cases of statin-related heart failure were published in 1990. 5 previously stable cardiomyopathic patients had a dramatic deterioration in myocardial function measurements and in clinical status shortly after beginning lovastatin. These patients returned to pre-statin condition after stopping their statin therapy and doubling their supplemental CoQ10 from 100 to 200 milligrams per day. So in 1990, they had these heart failure patients, 5 of them, take statins, and basically their myocardial function just significantly declined, and then it improved once they stopped, and then we're given CoQ10. So this is only 5 people, but you are seeing this in humans, and you are seeing this as a function of the statin, which is still interesting, even if it is 5 case studies. In 2004, it was demonstrated that diastolic dysfunction developed in 10 of 14 healthy hyperlipidemic subjects after 3 to 6 months of atorvastatin at 20 milligrams per day. Impairments in the ATP-dependent process of diastole. So diastole is the relaxing portion of the ventricles of the heart to allow for a filling of the blood so that the ventricles can then contract and push that blood out to the aorta and the other major vessels of the body. So what they're saying is there's an impairment in ATP-dependent process of diastole is an early finding in congestive heart failure. So what they mean by this is that ATP is necessary for the heart to relax. So a lot of people think that ATP actually provides energy for contraction. Actually, the high energy state of the muscle cell is relaxation. The ATP comes in and allows for the cell to be in a relaxed state so that you can have the contraction. Without the ATP, the cell stays in a contracted state, and this is what you see with rigor mortis, which is when the animal dies, and in death you essentially have a breakdown of energy production of the cell. Well, when the animal dies, now the muscles are all contracted up because they don't have any ATP generation. So this process of impaired diastole, or like an inability of the heart to relax, is because of a lack of ATP generation, which I guess to some extent is a spectrum, if you had a spectrum of life to death, it's like the heart moving closer to death. Like it's had decreased ability to produce energy. And so it's like moving closer to that rigor mortis process. I guess that's one way to frame it, at least. In this study, the early diastolic dysfunction was asymptomatic and reversed to normal after 3 months of supplemental CoQ10 at 300 milligrams per day, while the patients continued to take their statin therapy. So what they're showing here is that, and I'm just going to move this chair back a little. What they're showing here is that the statins are, or seem to, at least in some humans, 10 of these 14, the statins are leading to heart dysfunction, and CoQ10 is reversing that process. So the mechanisms that they describe above, where statins deplete CoQ10, as we discussed here, are at least bearing out to some extent in humans research or human case studies, where when you're giving people CoQ10, it's correcting their statin dysfunction. So that's just something interesting to keep in mind, that these mechanisms may not just be mechanisms, we're actually seeing it play out in humans. So as they continue, they say in 2005, 50 consecutive patients presenting with severe statin side effects were followed up for a mean of 28 months. In addition to symptoms of muscle pain and weakness, fatigue, dyspnea, peripheral neuropathy, and memory loss, roughly 1/4 of these patients had evidence of congestive heart failure at the time of presentation. All 50 patients had their statin drug discontinued due to side effects, and all were supplemented with an average of 240 milligrams of CoQ10 per day, followed up for 2 years. The patient's chief complaints improved dramatically, and 50% of those with heart failure showed significant improvement in heart muscle function. There was no adverse effects from statin drug discontinuation, with no myocardial infarction or strokes, and no side effects from the CoQ10. So essentially, 50 people had presented, 50 consecutive patients that presented with severe statin side effects were followed up for 28 months. They looked at their symptoms. Those symptoms included muscle pain, weakness, fatigue, dyspnea, peripheral neuropathy, and memory loss. Roughly 1/4 of the patients had evidence of congestive heart failure. So 1/4 of these 50, which would be about about 13 patients, 12.5 patients. So half of one of them had heart failure, so just one side of their heart. They had developed heart failure. When they discontinued the drug and were the statin and were given CoQ10, most of the patients' chief complaints improved dramatically, and 50% of the patients who basically showed up with heart failure had significant improvement in heart function. So if you had 12 or 13 patients, 6 to 7 of them actually, their heart function improved when stopping the statin. The other thing that they go on to say is that in that follow-up period, the 28 months, when stopping the statin, no one had developed a heart attack or a stroke or other side effects from CoQ10. So the side effects that they were having seem to be directly from the statin. They continue here another study. In 2008, a study in 29 patients with coronary artery disease found a significant increase in brain natriuretic peptide secondary to atorvastatin-induced plasma CoQ10 depletion after 3 months treatment with atorvastatin. Brain natriuretic peptide is a well-known marker for congestive heart failure. So in the hospital, we call this BNP. That's what brain natriuretic peptides acronym is. And what happens is the atria of the heart, when put under stretch, release this BNP, this peptide. And what the peptide does is it causes a loss of sodium and subsequently water in the urine. So that's why it's called natriuretic. So sodium's Latin name is natrium, and then uretic is short for diuretic. So it causes like a sodium wasting with water, or basically eliminating sodium. Why does it do that? Well, the atria are stretched, so the body is saying, oh, there must be too much volume on board, so let's get rid of some of this volume to take the pressure off of the heart. Or the pressure off the vascular system as a whole. And so BNP is a good marker for heart failure, because usually heart failure patients, their atria become stretched because their inability to effectively contract, or a high amount of vascular resistance, and essentially the vascular system is failing, so it gets waterlogged to a large extent, creates a stretch in the heart, and it can give us an indication on how bad that is. So what they're saying is that the depletion from CoQ10 from atorvastatin, which is a type of statin, led to an increase in BNP. So BNP being a marker of heart failure, would indicate that the CoQ10 depletion was impairing heart's function, leading to a buildup of fluid in the system, and then that leading to a stretch in the atria of the heart, and unable to effectively contract. So again, just an interesting. Now we're seeing like these mechanisms play out in humans. So statin cardiomyopathy, and we're almost done here. Yeah. So we only have one more page, I promise. What they discuss here is the mechanisms for the impairment in heart muscle function appears to be related to impaired mitochondrial function, which in turn related to statin depletion of CoQ10, selenoproteins, and Heme A, all required for normal mitochondrial function. We discussed this at length above. Statin-induced impairment in heart muscle function appears to be permanent, and even though patients may clinically benefit from discontinuation of the statin along with supplemental CoQ10, we believe that many years of statin drug therapy result in gradual accumulation of mitochondrial DNA damage. A prolonged decrease in mitochondrial CoQ10 would diminish the ability to protect mitochondrial DNA from free radical damage. After a critical percentage of mitochondrial DNA is mutated, offspring mitochondria will progressively lose their capacity to produce ATP and simultaneously can generate more free radicals and result in a self-perpetuated vicious cycle. So everything that we talked about before, I even mentioned this DNA mitochondria piece. The statins, so they're saying that the statin can lead to permanent heart dysfunction, which is actually quite scary. And the way they're discussing this is that blocking CoQ10 production and Heme A production and impairing antioxidant defenses over the long term with statin use impairs mitochondrial function that can lead to reactive oxygen species damaging the mitochondrial DNA. And as I talked about before, there's only so much repair that the cell can do because it's the cell or the body repairing itself. So if you damage a large portion of the body, even at the cellular level, it doesn't have the resources and equipment to then repair itself. So if you damage the mitochondrial DNA to a certain level, there's a limit to which it can be repaired. Once it hits that threshold, the next mitochondria that are produced from the DNA, if it's even possible for them to be reduced, can progressively lose their ability to produce ATP. Not to mention, if you're still taking the statin, you're still low in Heme A and CoQ10. Plus, you don't have all the antioxidant enzymes. So it's a progressive decline. They continue to say the negative consequences of statin-induced increase in coronary artery disease, coupled with the direct statin toxicity upon the myocardium, can be expected to be additive with enormous clinical implications. With more than 1 million heart failure hospitalizations every year in the USA, the rapidly increasing prevalence of congestive heart failure is now described as an epidemic, and it is likely that statin drug therapy is a major contributing factor. Last thing that they talk about, so statins are known to have a pleiotropic effect. So pleiotropic effect means they have these other effects that aren't direct to the mechanism that they describe with cholesterol lowering. So what they discuss here, and this is a side piece, a bit tangential to the overall argument, but it does play a part. So persistent inflammation is considered a major risk factor for atherosclerosis and the heart failure. Statins are known to suppress the prenylation of RHO protein and its downstream inflammatory cytokine production through nuclear factor kappa beta. So this is one of the major inflammatory signaling mediators inside the body. So it's actually like a gene segment that gets triggered, or like a protein that gets triggered and triggers a whole host of these cytokine productions. It's like a coordinating protein to some extent. Contrarily, statins decrease LDL levels, leading to increased entry of lipopolysaccharide into cells and increased inflammatory cytokine production. Thus, the effects of statins on inflammation is likely to vary depending on pathophysiologic conditions. So they're saying here that statins can decrease the nuclear factor kappa beta signaling, gene signaling, protein production, etc., by suppressing the prenylation of the RHO protein, which is involved in this cascade with of activating nuclear factor kappa beta and the other inflammatory cytokines. So it can have an immune suppressive effect, which can be good or bad depending on what you have going on, although immune suppression is usually not necessarily ideal, but that's fine. And then they also say statins, by lowering LDL, can allow increased endotoxin exposure to cells, which endotoxin is another name for lipopolysaccharide. I will get to this. I've discussed this on other podcasts before. I do want to go through some of the studies, but LDL cholesterol in general, binds endotoxin lipopolysaccharide in the cell, and in doing so protects the cell and tissues from lipopolysaccharide. So once the lipopolysaccharide is bound to the LDL, cholesterol, triglycerides in the bloodstream, etc., it basically inactivates it to a large extent and brings it to the liver to be removed or detoxified, etc. So blocking your LDL production can increase your risk of exposure to lipopolysaccharide and endotoxins. There's actually some interesting studies where giving people like cream or bile acids or rats like cholesterol protected them from sepsis by binding up the bile acids. Last thing they get over to here is that on the basis of these and other statin pleiotropic effects, statins seem to act as immune suppressive agents and may have beneficial effects in those who have excess and or life-threatening immune inflammatory reactions, such as in transplantations. However, immune suppression may be harmful in those who have no immune/inflammatory disease. Another thing that's immunosuppressive are omega-3s. I discussed this directly in my email newsletter in one of the studies. I will actually probably cover the study directly in a video as well because it's quite an interesting one. But it's essentially you don't want to be causing in certain situations where you have a transplanted organ, not having your immune system destroy that organ, like for example a liver or a lung or a kidney, is extremely helpful because usually when you have the transplanted organ, it's because your other organ has already failed or is not working. So you still need that organ to work. So sure, in that case, it's helpful. But for normal, healthy people, taking a statin to immunosuppress because you're under inflammation, if it has all these other mechanisms going on, like there's obviously other anti-inflammatories that are available that don't necessarily need to immunosuppress you for them to function. The other thing to keep in mind is that statins do have an antibiotic effect to some extent, so they can modulate the gut microbiome. They do have an antioxidant effect, which is known. So there are other effects that can explain some of the possible benefits seen with statins, but considering all these other effects that the researchers are discussing, I doubt that using statins over the long term is going to be a good idea. I would, from my perspective at least, I would probably look more towards finding other components or compounds or foods or strategies to help with heart disease, lower inflammation, modulate the gut microbiome than going with a statin. So overall, that's really it for this article. I know this is quite a long one. I hope you guys enjoy this type of content. As always, if you want to see me do more studies like this, go over more studies like this, if there's any studies that you want me to do specifically, you can go ahead and you can either throw them in the Telegram chat that you can pick up. I'll put in the description of this video. I also linked that Telegram chat in my email newsletter. And then you can also drop a comment here. And of course, if you like the video, go ahead and like it. And if you want to see more content, you can go ahead and subscribe. Hope you guys enjoyed the content, and I'll see you soon.